beginning. And I always was impressed how he used his authority in the field to push us, to motivate us, and especially to remind us that this was not an academic exercise or anything, but that this was really an urgency for many, many people in the world. And he has always made sure that everyone understands that we need to find cures, but also prophylactic vaccines for everyone in the world. So these things need to be soon, and they need to be accessible and affordable for all. And I think he will tell us, after the cure has been accomplished now, more or less, he will tell us now how we can get there for the vaccine side as well. Thank you, Michael Houghton, for being here.
Thank you very much, Georg. Can you see my slides okay?
Not yet. We saw them quickly before.
Oops. Let's try again.
Would you like me to try, Michael?
Yes, please, Lindsay. Thank you. Is it on full screen, Lindsay?
It is, yes.
Okay. Well, thank you, Georg, for your kind comments. Obviously, it would be very nice to develop a vaccine to Hep C, obviously. And I think if we do that, then we can put an end to this epidemic. So thanks to the organizers. So this is my title of my talk. What's the feasibility of delivering a vaccine this decade? Next slide, please, Lindsay. I apologize. Now I can't even see your screen. Are you there, Lindsay?
I am, yes.
I'm sorry, I can't see the slides now. Hang on.
We can see them, but
you can see them, but I can't see them.
But you can see them.
Oh my word. Okay. Thank you, Lindsay. So yeah, it's been interesting. This morning, some fantastic science presented by all the presenters, and there still seems to be a little bit of debate how important antibodies are. So I wanted to start with just some context on this topic. Next slide, please. So, you know, what direct evidence do we have for the protective role of antibodies? To me, the work that Dr. Mühlemann did, published in 2011, has impressed me a lot. So it's using the SCID UPA mouse model developed by my colleagues at University of Alberta, Lorne Terrell and Norm Kniedemann and David Mercer, in which SCID mice can be used to colonize human hepatocytes, and then you can infect these mice with Hep B, Hep C, and other pathogens specific for the liver. And so what Philip Mühlemann did was to infect these mice and give irrelevant antibody as a control, or he gave immunoglobulins purified from Harvey Alter's famous patient H. And these were antibodies purified from the chronic phase of H's infection. And Jane McKeating and others have shown that in the chronic phase of this patient's infection, there were broad neutralizing antibodies that were present. And so Philip asked the question, what happens if we passively immunize these mice with this purified immunoglobulin and challenge with homologous virus on the left of this panel? And you can see very clearly relative to the control antibody, which was the same just a control antibody isotype, that the HIG protected these animals very well, shown in red. After 2 weeks, there was no infection registered in these animals. Then he went on to challenge with heterologous virus, 4A and 6A. And at week 1, all the control antibodies were infectious with high viral loads. But in the animals that received the IG from H, as you can see, 6 animals were negative for viral RNA. The important thing to remember in this model is that the half-life of human IG is very short. It's only in the region of around a week. So in subsequent weeks, with regard to the challenge with 4A, 2 of the 6 mice were still negative. The other 4 were positive, as you can see. But even so, some of these animals had less viral load than the control antibody mice. And even in week 3, as you can see, there was 1 mouse that was still negative for viremia. And pretty much the same pattern when he challenged these mice with 6A, a very diverse genotype. On week 1, 3 of the 4 mice with the HIG were negative, unlike the 3 control antibodies. So this has always impressed me regarding the preventative role of immunoglobulin, given that the half-life is very low in this animal, very short. Next slide, please. And also I was impressed by this work from Dr. Moran and colleagues in 2012, where he used the chimpanzee model and then passively immunized with the HCV1 monoclonal antibody. The control is shown in green, was not passively immunized. And as you can see in green, the animal quickly became viremic after challenge, experimental challenge. In red was an animal that received the monoclonal antibody at 50 mg per kg. There was obviously viremia, but when he used a higher dose, 250 mg per kg, shown in the animal shown in blue, as you can see, there was no infection. So this is a broadly neutralizing monoclonal antibody, and when it's there, it does its job. It prevents infection. So I think these are a couple of examples. There are others. Manson published data in the mouse using broadly neutralizing mAbs to show that they were able to protect mice as well, the UPA mice. So I think it's important just to remember that there is direct evidence for the protective role of antibodies. Next slide, please. And I guess I've used the chimpanzee model to test HCV vaccine candidates more than anyone else. When I was at Chiron company in California, we used it a lot with different types of vaccine. One of them was a T-cell vaccine. It was a non-structural protein, polyprotein, that we adjuvanted with Iscomatrix, a very powerful adjuvant. And in chimpanzees, we saw a very nice amelioration of acute hepatitis and acute viremia. But when we followed 5 of those animals up, only 1 actually resolved the infection, which was actually a lower rate than the control animals, where 3 out of 5 resolved approximately. 2 out of 5 resolved. So, you know, pretty much it seems to have happened with the Ocuris vaccine. It elicited strong T-cell responses, CD4 and CD8, and yet there was no effect on persistence. And that's exactly what I saw with our version of a T-cell vaccine in chimpanzees many years ago. But when we tested adjuvanted recombinant E1 and E2 heterodimer in the chimp model, we saw a very different picture. And I regret not publishing this data in more detail. We put it into a couple of reviews, as you can see here, 2005 and 2011, with yet more data. But I stand by this data. These animals were very carefully monitored by Amy Weiner, and who was a very thorough scientist. And we're monitoring chronic infection, obviously, by the detection of viremia using PCR. And so we immunized these animals, naive animals, with around 50 micrograms of E1E2. We did use different adjuvants throughout the study. This was done over several years. We could never, even in industry, we couldn't get enough money to do all the animals in one time. So there were various adjuvants used with the E1E2. But as you can see, with the homologous challenge animals, 12 of those that were challenged, 5 were completely sterilized. No viremia in blood, PBMCs, or in the liver at any time. And these were the ones that had the highest antibody titers. Now, unfortunately, at that time, this was many years ago, we weren't able to monitor T-cell responses to the vaccine. But at least we did see some sterilization. And in the 7 animals that became acutely infected, when we followed these up, only 2 became persistent. Whereas of 10 unimmunized animals, 7 of them became persistent. And there is a statistically significant difference in the carrier rate. Then, as you can see, when we followed up this work by challenging animals with a heterologous H77 isolate, so our vaccine was made from the HCV1 isolate, which is a 1A, when we challenged with a heterologous 1A in the form of H77. We didn't see sterilization. All 19 vaccinees got acutely infected, but when we followed up, only 3 remained persistently infected, unlike 57% of unimmunized controls. So once again, a statistically significant difference, and put all the data together and it's highly significant. So, you know, unfortunately, we weren't able to do more challenges with different genotypes, but I think, to my knowledge, this is the only Hep C vaccine candidate that has been shown to reduce the incidence of chronicity and viral persistence. So that's why I've stuck with E1E2 for so long. Next slide, Lindsay, please. Thank you. So on the basis of this chimp data, the NIAID co-funded a phase 1 clinical trial. The vaccine was obviously made at Chiron. And we used MF59 adjuvant and oil in water emulsion. And we immunized individuals with different doses, either 4 micrograms, 20 or 100 micrograms, given 4 times. And to summarize, we got a satisfactory safety and tolerability profile. We saw anti-E1E2 ELISA titers in the same range as in the protected animals. And this is work by Sharon Fry, who conducted the phase 1 trial in St. Louis, and Ranjit Ray did some neutralizing antibody assays at that time. Most vaccinees elicited significant titers of neutralizing antibodies to the heterologous 1A pseudoparticle. We saw very strong lymphoproliferative responses to E1E2. Mainly those were CD4 T helper responses. And importantly, E1E2 is a very difficult protein to express and purify, as probably some of you know full well. The good thing about this trial is basically 4 micrograms is pretty much optimal for antibodies and for T-cell responses, lymphoproliferative responses. So that's very important, because although E1E2 is difficult to manufacture, a moderate scale up by industry standards would be something like 1,000-liter cumulative bioreactor volume. And at the yields we've been getting, we have two methods to purify it, but using the most efficient method, this level of bioreactor would yield enough antigen for somewhere between 500,000 to 1 million recipients who would receive 3 doses each of 4 micrograms. So, yes, it's relatively low yielding in a industrial vaccine world, but it still can provide enough vaccine, I believe, especially for the high risk groups who are the people who inject drugs. Next slide, please. Yeah, so a lot of great work this morning was presented on the issue of cross-neutralization. And I certainly accept the points that Justin and Manson were making. I sort of have a little bit of a different perspective. And I guess my perspective is that this wild type E1E2 from a single strain doesn't have a bad cross-neutralizing profile. And I would make two other points. One is that traditionally the vaccine world, the corporate world, deals with heterogeneity by combination vaccines. For example, pneumococcus vaccines have now, I believe, 30 antigens from 30 different strains. In the viral world, we've known for decades that poliovirus vaccine is a trivalent vaccine comprising the 3 serotypes. Papillomavirus vaccines comprise 4 different serotypes. So I think the work that was being reported this morning is fantastic and really, really excellent. And it obviously could yield very good improvements to our vaccine antigens. But let us not forget that the traditional way to deal with heterogeneity in commercial vaccines is combinations. So if we use the combination, for example, of genotype 1 and 2 and 3, I take Justin's point, we won't neutralize all the serotypes within those major genotypes, but we will get a pretty good coverage. And the second point is that nobody knows what level of neutralizing antibodies we need in order to be protective. I think that came over loud and clear this morning. And so let us not talk ourselves out of testing available vaccines now for efficacy. I think that's my message. And very quickly, I'm running out of time as usual. This is old work from Jane McKeating's lab in guinea pigs. And what she's showing is cross-neutralization of 1A in black. These are different guinea pigs. Cross-neutralization of 1A in black, 2 1B strains in the stippled and in the clear bars. And even against 2A, the J6 strain, she saw significant but reduced cross-neutralization. And next slide, please. We gave our vaccinated chimpanzees to Bob Purcell, and in his lab, J.C. Mounier and Jens assayed for neutralizing antibodies. They did CC as well as PP. We gave them 4 chimpanzees. And so you're looking at the neutralization from those anti-sera against all the major genotypes, 1 through 6. And what they found was, pretty darn good cross-neutralization for most of them. Genotypes 2 and 3 were less well neutralized, but they're still partially neutralized. And we don't know what levels of neutralizing antibodies are needed for protection. So I certainly agree that we should try and optimize cross-neutralizing antibody profiles, but let us not forget, these might be sufficient, especially if, as we are doing, if we are going to make combination vaccines of genotypes 1, 2, and 3 to test in humans. Next slide, please. And then John Law, my colleague at the U of A, University of Alberta, as you know, he published some data with the human volunteers that got MF59 adjuvanted E1E2, and we're only showing 3 of the volunteers here, but we saw a profile somewhat similar to what Bob Purcell found with our chimps, a broad cross-neutralizing profile. Given that this is just from a single 1A strain, this is pretty remarkable, I thought, and still do. Genotypes 2 and 3, once again, were not as well neutralized in the human volunteers as the chimps, but historically, we've solved that issue by adding making combination vaccines. Next slide, please. And then one of our students, one of our excellent students, we had two excellent students, Janelle and Jason, and Janelle published a paper a couple of years ago showing in goats, we immunized goats to get a lot of anti-sera so we could do more. We didn't want to use the precious human anti-sera. But Janelle showed in the goat model, at least, she was able to get very good neutralization of JFH1. J6 was not well neutralized, but JFH1, this certainly confirms what Justin was saying this morning, that neutralization is not necessarily genotype dependent or subgenotype dependent, but at least it shows this E1E2 can neutralize a lot of different genotypes and a lot of different subtypes. Next slide, please. Next one, please. Yes, we had some discussion yesterday that Stephen rose in discussion. Obviously, very nice work from Jens and other people showing that if you remove the HVR1 region at the N-terminus of E2, the virus becomes very susceptible to neutralizing antibodies. And so the hope was if we remove that from our E1E2 molecule, perhaps we can increase antigenicity. But next slide, please. In terms of reactivity with BNAb monoclonals, we saw a reduction in activity to the top three antibodies. These are, I believe, broadly neutralizing monoclonal antibodies, including AP33. The deletion actually reduced immunoreactivity, but a lot of the other good BNAb monoclonals, 8426, 3B, 4A, 5A, we saw no difference. But what that, next slide, please, what that translated to in terms of antigenicity is shown on this slide. We immunized guinea pigs, and then we tested for cross-neutralize. Well, first of all, we tested for neutralization of homologous virus, H77, and you can see that the deleted E1E2 was not as good at neutralizing homologous virus as the wild type, undeleted. On the right-hand side, you see that they were pretty similar in terms of neutralizing HK6A CC virus. So overall, we didn't really see an improvement by deleting HVR1. Next slide. So we've gone ahead and we've made E1E2 from strain H under GMP. I believe when it's combined with perhaps genotype 3 E1E2 and perhaps genotype 2 E1E2, we will get broad neutralizing activity. It may not be perfect. I agree with Justin and Manson, but it may still be effective. And anyway, I'm showing you here the GMP produced E1E2. In track 1 on the left is a silver stain, and that's showing E2 alone that was secreted. And it's the ectodomain from 384 to 688 that was purified. Track 2 is one of our GMP batches. And you can see E2 and E1 fairly clearly. And then in track 3, we've got sort of the old Chiron vaccine, E1E2, that's been purified with GNA lectin. Once again, you can see the E2 and the E1 pretty clearly. And then track 4 is another batch of GMP produced material from University of Alberta. And then the Westerns are shown on the right-hand side. The E1 is obviously lighting up very well, as well as the E2. So we've got pretty good GMP material now. Next slide, please, Lindsay. And what we've done with it is used Manson's and Stevens' very illuminating monoclonal antibodies. Most of these are broadly neutralizing monoclonals. They neutralize most of the genotypes quite effectively. And this is interesting because it shows that the E1E2 batches shown in red and blue are reacting with these monoclonals, binding to these monoclonals better than E2 alone. You can see that with the top 3 antibodies, AR1 through 3A. And then on the bottom are Manson's AR4A and AR5A monoclonals, which depend on both E1 and E2 for binding activity. And you can see we get very good activity of our 2 GMP batches, equal in binding to the, if you like, the chiron-like E1, E2 that we tested in phase 1. And importantly, there's no binding of these important monoclonal antibodies to the E2 alone antigen. And then the next slide are some of the results with Stephen Fong's antibodies. His 205 and 210 shown on the right-hand side are binding in the same region as Manson's AR4 and AR5 regions. And we saw the same picture. The E1, E2 GMP material binds very well, but the E2 does not. And on the left, one of Stephen's antibodies, HC84, which is binding in the AR3 region, as defined by Manson, is binding to the E2, but not as well as against the E1, E2. So I think this is important because, in my opinion, E1, E2 is the best antigen to use to elicit cross-neutralizing antibodies. Next slide, please. As I'm saying here. And of course, with the fantastic work done by Oxford and AstraZeneca and BioNTech and Moderna, we now have the option of delivering these antigens through mRNA or through viral vectors. Next slide, please. It turns out that a group at Novartis back in 2015, this was the group of Andrew Giel and Dr. Brito, published actually 7 years ago, surprisingly, in the results of immunizing mice with 10 micrograms of self-amplifying RNA encoding E1, E2 heterodimer. And they compared it with immunization of mice with 1 microgram of my adjuvanted E1, E2 protein that I produced when I was at Novartis and Chiron. And well, it's an initial experiment, but I think what it shows here is the inhibition of neutralization. You can see that the protein, the 1 microgram of protein, seems better than the 10 micrograms of self-amplifying RNA. But given that this is the first attempt, I think it's telling us that we can deliver E1, E2 through either self-amplifying RNA or through mRNA, and I'm sure through adeno-based vectors as well. Next slide, please. So my feeling is we have many options now available for developing a Hep C vaccine. I am very optimistic. We have the protein-based vaccine. I personally think it should be E1, E2 heterodimer. It can be enhanced the old-fashioned way by making a combination of antigens from genotypes 1, 2, and 3. I think we will have a pretty good cross-neutralizing profile if we do that. Justin will argue quite rightly that it can be improved, but I think it's not bad. We can scale this protein up, and of course, if you deliver it by mRNA and vectors like adeno, it should be a relatively easy scale up. We are, like many groups that I've heard at this meeting, we're in addition planning to dial in T-cell responses. What we're doing is a bit different from Okiris, for example. We're focusing on highly conserved epitopes. And to do that, we're incorporating synthetic peptides to highly conserved T helper and CD8 epitopes. And we're using a cocktail so that we can cover many MHC alleles so that we can hopefully induce T cells, CD4 and CD8s in most of the human population. We need to use a good adjuvant. My colleague, Dr. Amir Landi, has done a lot of work in the mouse looking at different adjuvants. We're impressed with the data from Bali using the TOL7/8 agonist adjuvants. We've been looking more at TOL4 agonist adjuvants, and we've been impressed, certainly in mice, with the strong responses, both CD4 and CD8, that Amir's been getting with E1E2. And I'm actually encouraged by a paper from Adam Penn Nicholson in Vaccine 215. They had a fusion protein made from TB, tuberculosis, and they used AS01, this GSK adjuvant, and they showed in humans that they could get very strong CD4s and CD8 responses to that antigen. And as I said, we've got the mRNA and the viral vectors now to deliver these antigens. Next slide, please. So my belief is that we can roll out an effective Hep C vaccine, at least to the people that need it the most, who are the people who inject drugs, this decade. I believe that's very feasible. I also believe, as you've probably gathered from my talk so far, that I believe the wild type antigens that we're using are more than worthy to continue clinical development. I think there will be multiple phase 1 trials, I'm sure, reported over the next 1 or 2 years from many groups. And if we get good, reasonable cross-neutralizing antibody profiles, if we get cross-reactive CD4 and CD8 T cell responses, and if the CD8 responses are presented by multiple MHC alleles, then I think we should proceed to efficacy testing. Now, Jake Liang organized a very nice meeting last year at the NIH to discuss human challenges, and I think this is a good way to go. We are talking to regulators in Health Canada right now about how to do these trials, and I do think, I agree with Jake and colleagues that this is a good way forward. So I expect, hopefully, to be seeing some efficacy reads out in humans in 24 or 25. And I think if we can show that volunteers that are vaccinated and then challenged actually go on to develop a very low chronicity rate, that compared with all the studies, historical studies, where we know what the chronicity rates are for lots of different populations around the world, and they're very high, as we all know, I believe that should allow us to start rolling the vaccine out in a staged, sequential way to people who inject drugs. And then we can follow up larger numbers of those individuals to confirm that the chronicity rate is a very low level. And then after that, I'm sure the brilliant work from Manson and Justin and everyone at this fantastic conference has talked about, we will, I'm sure, get a superior vaccine emerging over the next few years. And then we can start dialing that in, of course. So next slide, please. I think it's my last. Yep, just like to thank all the many colleagues I've had at the U of A, the research team of John, Mike, Amir, Judica, Darren, Jason, and Janelle. We have a large GMP team that are really doing heroic work. E1E2 is not an easy protein to make. My dear friend and collaborator, Lorne Terrell, has supported us all the way. And would like to acknowledge that this work started many years ago at Chiron with my co-discoverers of the virus, Quilin Chu, George Kuo, and then also involved, the vaccine work also involved Bob Ralston and Richard Spady. Manson's and Stevens' antibodies have been very, very important in our interrogation of E1E2. Thanks to them for providing those antibodies at all times. And I'm really enjoying the U19 team that I'm on with Stephen, Bally, Chris, Richard, and Francois. and my colleagues at the U of A. So thank you very much, and happy to take any questions, and I guess we'll also have a discussion period coming up.
Yeah, thank you very much, Mike. This was great for giving a bit of a different perspective. And I don't have a question right now online, but